Our first speaker is going to be Anya van der Plas. She is essentially the Namibian oceanographic expert, and she's going to give us a presentation on looking at this spatiotemporal variability between nutrients and oxygen in the northern Benguela upwelling system. So Anya, as soon as you're ready, please go ahead.
Thank you, Shiba. Yes, so I'm going to talk a little bit about the variability both of oxygen and nutrients in the northern Benguela upwelling system, specifically focusing mainly on the shelf region. So looking at hypoxia, remineralization, also source water masses and nutrient fluxes, but not too much on the physical side, because I believe Hari and also I know Natalia last week also gave some information already on oxygen and nutrients. But I wanted to start off in saying, being from the Ministry of Fisheries and Marine Resources, we have regular environmental monitoring that we do, focusing on physical, chemical, and biological data. And survey lines you can see here. The 18 South is one of our monitoring lines, the 20 South. But most of the data I'm going to show will be from Walvis Bay region, which is our primary monitoring line. Although we do have some lines in the south, also the yellow one covered by the Ludwig's colleagues. So, but today my focus will be on oxygen nutrients. And here in the picture, you can just see our main research vessel, the Variability. The smaller vessel is the one that the Ludwig's colleagues use. So why monitor dissolved oxygen? First of all, it's fundamental to the marine biological, ecological, and biogeochemical processes. And thus it is critical to our life in the oceans. And decline in oxygen affects many things: the productivity, the biodiversity, and the biogeochemical cycles. If you look at the schematic, you'll see climate change, how it impacts through temperature or winds, the physical, environmental, also biological responses. And then here you see I've circled bottom water hypoxia. And similarly, here's anthropogenic activities showing with nutrient loading and nutrients being enhanced, but also through biological responses. You're looking at sedimentary carbon and nutrient pools, and that is sort of a little bit the other thing that I'm talking about. And we have seen that over the past 50 years, oxygen has decreased by 2% in the global ocean, and the volume of oxygen depleted waters has grown more than 4 times. And there are over 500 low oxygen water sites which have been identified around the world. So looking at deoxygenation, what processes are leading to deoxygenation of the ocean globally? First of all, there's of course nutrient enrichment, what we say eutrophication. A lot of this is anthropogenic, whereas in the Namibian upwelling system, the Benguela upwelling system, our nutrient-rich environment, it's all actually natural. It's not really through anthropogenic influences. Then you have increased greenhouse gas emissions. You also have, with global warming, you have rising temperatures in the ocean, which has an impact on, because it decreases oxygen solubility, it also leads to increased oxygen consumption. Then with these rising temperatures, you also have reduced ventilation. Your currents are becoming more sluggish in some instances, and you have enhanced stratification. So what are these effects that hypoxia has on fish? So hypoxia is when you have the low oxygen water on the sea floor, and also in the oxygen minimum zone off the shelf. So first of all, there's mortality. And here I've just shown a slide of some coastal mortalities that we experience. Here you can see rock lobsters that have been washed up on shore. And what happens near the coast is you have when you have a decrease in temperature, the blue here, you get nutrients put into the surface layer. Then as temperatures increase, you get stratification, you get chlorophyll blooms, that is the green here. And then when you have these blooms, they can be harmful algal blooms or just generally in the oxygen, and that's when you have low oxygen. It can even be so severe that you have sulfide eruptions happening, so sulfide events, and that's when you have mortalities of the local reef or surfstone species. Other effects of hypoxia on fish are that you see reduced growth rates. Hypoxia can also lead to altered distributions of the fish species and also altered behavior. For instance, that the fish lift off the bottom. Here I have an example why we do spatial monitoring, why we go out on our resources surveys. This is an example of the Hague survey that we do. On the left, the map with stations and trawls. And this is the Melusius capensis densities mapped with the low oxygen. And you see the red here, January, February is our Hague survey time when we have a lot of low oxygen on the shelf. So the red is less than 0.5 milliliters. And you see that the black dots is the Hague densities, that they generally occur in highest densities outside of this very low oxygen area. But you'll have, for instance, behavior that the smaller prey might be the smaller hake might be slightly more inshore, trying to hide away from the adult fish, you know, to avoid prey or to avoid cannibalism and things like that. So with our regular monitoring that we do, I mentioned the Walvis Bay transect, where we have our primary monitoring line. We have also developed what we call an oxygen index. And here you look at specific threshold oxygen levels. For instance, here in the red, it's a 0.5, which is sort of been shown as physiologically the critical oxygen concentration for hake occurring. Whereas the yellow area for that is sort of the lower threshold, 2.5 milliliters per liter oxygen that we find for some juvenile pelagic fish species such as sardine. And here we've mapped over time how large are these low oxygen areas, and the reverse is basically how small is that in the central area, the habitat becoming seasonally and also through interannual variation, the variability for the fish to occur. Because you can see here 2000, 2001, for instance, you see a prolonged low oxygen event. That means that the area was really, for some juvenile fish at some stages, very small, also even for the sardine off the central coast. And then the fish would react with either moving offshore, or you would have effects, for instance, with the recruitment of the fish. Yeah, so other effects of hypoxia on fish are also altered food webs and trophic interactions. And of course, you have multiple stressors between the mortality and the distribution, and that reacting together was having a large effect on the ecosystem. And this problem of deoxygenation is predicted to worsen in the coming years under continued global warming and increased nutrient input to coastal regions as human populations and economies grow. So here I just show again the Walvis Bay long-term monitoring just for a particular station that we have, which is the 10 nautical mile station. It's a relatively shallow station, just 100-meter deep. But you see here from 94 till 2017, there's a lot of variability. And this low oxygen water variability results from complex interactions over the shelf. There's the remotely forced shelf processes from the Angola basin. I know Natalia talked a little bit about that. There's the weakening of the upwelling winds, which will affect the low oxygen water distribution. There's also the seasonal thermocline variability. And then, of course, the biogeochemical carbon fluxes on the shelf play a large role. The lower graph here, I actually show that same time series of dissolved oxygen, but showing it as oxygen anomalies so that it shows up much more clearly when do you have periods of longer fluctuations and stronger fluctuations in oxygen. For instance, here in 96, you see the red, you had ventilation, strong ventilation event, whereas here in 2000, 2001 is where you had prolonged low oxygen water, prolonged hypoxia over the shelf. And by showing it as anomalies, you see this showing up a lot more clearly. And we see that there's definitely a lot of interannual periods of longer term variability in the oxygen over our shelf. And these seem to be forced by recurrent warm and cold water events. So just looking briefly at these remotely forced effects. Here you see on the left hand the Eastern Tropical South Atlantic system. We see here the Angola jar, and here is the Cape Basin of South Africa. And you have shelf hypoxia arising seasonally because of the advected hypoxic water here, which is nitrate rich, which is the South Atlantic central water from the Angola jar. This is usually summer to autumn. And you can see here on the right here, this pool of low oxygen water that sits in that Angola jar, which comes up here, especially off northern Namibia, where it upwells onto the shelf seasonally. Whereas in winter, you have seasonally advected oxygen-rich water, which comes from the Cape Basin pool, which is the Eastern South Atlantic central water, mainly during the upwelling season in spring. And that you see here with this very much higher oxygen that you have in this pool of water. Generally off Lüderitz is where you see this upwelling onto the Namibian shelf. So I've alluded also, besides the oxygen here, to the nutrients, and I know Natalia has also talked a little bit about this. So let's have a look briefly now at the macronutrient. We started collecting them in 1995, end of the year, and here when you look at nutrients, here you have ammonia, just the general distribution, nitrate, phosphate here, and silicate. You see that most of the variability and also the highest nutrients are in the upper about 200. So let's just have a look at the sediment and the benthic boundary layer, and this information is from off the Walvis Bay transect of central Namibia. Here on the left, you see pore water, ammonium versus phosphate, and we see that the relationship is very far removed from the Redfield, which is the dashed line is the Redfield ratio of 16:1. So we see it's very much removed. And if we look here at this middle graph, which is inshore ammonium versus phosphate, we see that it's a low entropy ratio. So this informs us that from at the inshore stations, you have an influence of the pore water ammonium flux from sediments to the benthic boundary layer here. Whereas if you look at the offshore graph here at the bottom, you see that there's no real relationship, and offshore the benthic boundary layer is decoupled from the sediment flux. If we look at nitrate versus phosphate relationship, we have inshore, we see it's more of a negative relationship, you see that whereas offshore, it's much closer to Redfield, although it's still a lower N to P ratio. And this is where the system further offshore from about 30 nautical miles offshore, where it switches to more aerobic conditions. So just briefly looking at surface nutrient concentrations we have on the top here, we have silicate versus phosphate, and we see that there is a clear seasonal pattern in nutrient concentrations with high values during winter to spring and lower concentrations offshore versus the inshore area. And the seasonality in the, we also see in the surface layer, also higher in winter versus in spring, or sorry, surface layer, this is for nitrate, but we also see that here there's a little bit more uniform distribution. Looking at the bottom nutrients, silicate on the top and phosphate on the bottom, there's not really that seasonally distinct pattern, although we do see that for silicate we have stronger, higher concentrations inshore versus the offshore. And if we look at nitrate, we see a very strong gradient, much higher nitrates offshore than inshore. And we see that there are very low nitrate concentrations inshore during the summer to autumn period. And this is due to nitrate reduction processes such as denitrification and anemox mediated by bacteria near the sediment water interface. So looking at the nutrient cycling, basically what you have is a nitrogen source or nitrogen excess, which arises here in the Angola Jaya region seasonally, and this is transported onto the Namibian Upwelling shelf, the northern Benguela here. But here off Namibia, you have the formation of a nitrogen deficit on the shelf due to these aforementioned nitrate reduction processes, also due to sedimentary efflux of phosphate, and then also the advected source waters that are on the shelf. And this leads to basically a phosphate excess that you have, a phosphate exported here from the Namibian shelf into the subtropical South Atlantic. So finally, just touching on briefly on some regional projects. There's, together with some institutes in Germany and also South Africa and Angola, we have a traffic project looking at trophic transfer efficiency in the Benguela current. And one of the work packages here is looking specific at carbon or energy transfer at the lower food web in the northern and southern Benguela upwelling system. So here we are looking at carbon and nutrient cycles. I don't want to say too much about that because Tibo will be talking about carbon cycles in the next talk. And then also I just briefly wanted to mention the VOICE project, which is variability in the oxycline and its impacts on the ecosystem, which is an international demonstration project focusing on oxygen minimum zones and the effect the spatiotemporal, the changing oxygen minimum zones, how they affect the spatiotemporal distribution, productivity, and trophic structures of the benthic and pelagic communities. Here specifically focusing on the upper oxycline. And also linked to this, a separate group where I didn't put in a slide is the GON group, Global Oxygen Observing Network, where they are also looking at decreasing, declining oxygen levels worldwide, and there have been some publications and a new one, Pitcher et al., which will come out soon. So that is it. Questions, please? And I hand over to the panel.
I see there are 3 questions for you. One from Brahman. Maybe Brahman can rephrase the question. It's in shorthand.
Anya, it was interesting to see the distribution of the hay. There were big concentrations sort of on that oxygen limit between the severe oxygen depletion to slightly higher one. Do you know why?
Sorry, Bronwyn, that there was severe oxygen, this slide here.
Okay, so presumably large circles, are those Haight catches?
It's the densities here that's being depicted, but that is where the largest occurrences of capensis.
Okay, so it's quite apparent there that the largest densities are just sort of on that borderline between the very low oxygen and the higher one. My question is, do you know why? Is it simply that they're avoiding the low oxygen, or they're combining the food abundance to be there?
I think it's very much that they're at the edge because they're avoiding that low oxygen. They would come in periodically, but of course it depends on the thickness of the layer, which in this map it's the bottom oxygen, but it doesn't give exactly how deep, how thick that layer is, because the vertical extent also varies, of course.
Okay, thank you, Bronwyn. Yeah, I think your question is interesting. It might also be partly due to that reason. There are two questions from Sam. Maybe he can answer them himself, but they are quite clear. It's in the comments. Anya, what's the difference between anoxic zone and the dead zone? And does Namibian shelf qualifies as a dead zone? Those are the two questions from Sam, and his connection is not that stable.
Yeah, I think Bronwyn is probably more qualified to talk about that. But anoxia basically refers to zero oxygen, whereas hypoxia, the term varies. Generally, it depends if you're a biologist or if you're in oceanography. If you're talking about hypoxia, it's below 2 milliliters per liter oxygen, up to about, say, 0.5. If I talk about hypoxia below, so 0.5 to 2 milliliters, mainly 0.5, but anoxia is below that, basically zero oxygen. And when they're talking about dead zones, it's basically above the seafloor, that area where you generally don't see organisms coming because there's no oxygen there. But that is also, again, wrong to say dead zone, because you do get some organisms that have their that survive by using other chemical reactions other than that can survive anaerobically.
Thank you very much, Anya. I don't see any questions in the chat box, but if there are any others that would like to ask a question, please do so.
I have a question. That's okay.
Yes, go ahead.
So the depletion of nitrate relative to phosphate that happens in this area, and you showed some nice data showing that you've got really high P relative to N in the anoxic pore waters.
Yes.
Is that driven by a preferential release of phosphate from the sediment, or is that purely due to denitrification and anomox, or a mixture of both?
It's a mixture of all of those mentioned. You do have a release of phosphate in the inshore, in that mud belt zone, from the sediment. We saw that with pore water measurements, but also I think there's a Goldhammer paper out about the phosphate release. But there's also denitrification occurring, and also anomox occurring above that in the water column. And that's why it's a combination of that. But then if you look at MICETL, despite all of these processes happening, and you see that phosphate excess in MICE, they've modeled it to say that it's very much also the transported water mass properties of the aged upwelled water that has a big influence on the Namibia being basically a phosphate source. So that is very much, it's very much also a seasonally dependent on which water mass you're having on the Namibian shelf, and also if it's that low, so if it's that low oxygen period or not, and if it's the tropical water versus the Cape Basin water that you're having on.
Yes, go ahead. Good.
when I overlook your graphs, Anja, I feel that the yearly variability is so high that it's hard to see a trend. In other words, the 2% decrease in oxygen due to the elevated temperature is not really making a big difference. Or in other words, it has always been anoxic in the deep waters of the shelf. Or do you see a trend?
I'm actually very glad you asked that, Kurt. From my work that I've done, and if you see here, especially that's why I was doing these anomalies in oxygen, you see a lot of interannual variability, and it seems to be linked sort of more the Benguela Nino, you'd have more hypoxic conditions, whereas cooling events, sort of almost a Benguela non-linear type event, you'd have more ventilation events occurring. But I do not see on the Namibian shelf that there's a deoxygenation trend. I do think I should maybe look at if there's a shoaling. I haven't really looked if there's maybe a shoaling of the oxygen minimum zone, or maybe looking more on the off-the-shelf trend. But for me, I just see a lot of variability. But also, if you go back to some of the sediment core work that's been done, where they reconstruct oxygen time series, Namibia has always had this lower oxygen area.
Thank you, Anja. Any more questions for Anja?
Yes, from me, Chris.
Okay, Paul. Good.
Thank you, the moderator. Anja, the phosphate release that you have shown, I would like to believe that that is natural, or is it due to any kind of bottom disturbance?
No, this is all natural, Paul. This is to do with the sediment water fluxes, with the poor water fluxes on the shelf. And also, I think when Jake and them come in with the bacterial talk, there's a lot of bacteria that have adapted and that use these phosphates. So the whole microbial bit is not my field. I think you must listen in about 2 or 3 weeks' time to their talks to get some answers on that. But it's the stored phosphate, and that's why all this phosphate mining, there's this interest to do the phosphate mining of Namibia, because of these pools from our the high productivity and all of this organic matter on the production that sinks onto the sediment and these mud wells. They are so nutrient rich, and that is where a lot of these phosphates come from. Okay, I think time-wise, I think I should end here with my talk.